Thank you very much for the wonderful introduction, and thank you all for coming and inviting me to give this talk. And hopefully this will be a nice introduction to what bubbles can do and some of the work that I've been doing recently, mostly conducted in Singapore, but what I hope to be continuing doing here. So really quickly, I just wanted to emphasize that nature really doesn't like bubbles. And to illustrate this, I basically took a very short video of a bottle of water and I shook it. And you can know that nature doesn't like bubbles because when you shake bottles of clean water, bubbles don't persist. They immediately disappear. So here's just a quick video of that. You can see the bubbles are all present in our system, but immediately, I mean, this is slow motion, they seem to all disappear. Actually, with some slight exceptions, because the video didn't last very long, where you can kind of see some little tiny bubbles here, but I'll address that afterwards. So what's happening? Well, here we have initial state, which is a system in equilibrium, so it has a pretty much low energy in the system. I shake it by therefore imposing energy into the system, and I seemingly am able to create all these bubbles. And then I reach its sort of peak highest energy where it's in the least amount of equilibrium. And then over time, you can see that the bubbles seem to go backwards and return back to its original state. So you add energy, it has a peak amount of energy, and then of course nature prefers to minimize energy. That's based on the second law of thermodynamics, and we go back to our original equilibrium state. So why are bubbles so energetic? Well, it has to do with creating surfaces. The more you create surfaces, there's an energy involved, there's work that's required to create that surface. And that's somewhat proportional to the surface area, in this case of a bubble, so of a sphere, so it's roughly unit length squared. And so what nature tries to do is reduce surfaces by either coalescence. So here you can see maybe a bubble might have split. So coalescence would be these two bubbles recombine to make a bigger bubble. There's phase separation. So as a bubble reaches the surface of the liquid, you notice that all of a sudden it seems to join back to the air that's on top of the water. And there's another process that you can observe, which is Ostwald ripening, as in you can't observe it in this video, whereby molecules of a smaller bubble, the air that's in the smaller bubble will diffuse into a bigger bubble because that's more energetically favorable. And again, these bubbles will then rise, touch the surface, and then you get your phase separation. And I said earlier, you could kind of see some smaller bubbles that were present at the end of the video. But it turns out that nature really hates these small bubbles because the amount of energy per unit volume of a bubble is proportional to 1 over R. And what this suggests is that the smaller the bubble, the more energy it has relative to its volume. So that's why bigger bubbles tend to persist longer than smaller ones. And you can actually observe this because with smaller bubbles, they actually are not affected by gravity as much. They tend to sit in solution. That's why when I shook that bottle, you can kind of see them floating around. They don't just immediately float to the top of the surface. But it turns out that if you look at the rate at which, or if you look at the molecules within this bubble, there's so much energy on the surface pushing into it that it actually pushes, in some sense, the gas out of the bubble and into the solution almost as a dissolved gas, so not as a collective. And this is proportional to the radius, a term called surface tension, and then another term, which is how saturated the liquid is. It turns out that, of course, this term and these two terms are always positive. So for all values of F that are less than 1, you will always have a decreasing surface or a decreasing radius. And so this is actually a video that I took many years ago where you have a bubble in a saturated liquid, a saturated gas liquid, and I'm just watching how it changes with time. And what you can see is that over time, it's shrinking, it's shrinking, it's shrinking. And actually the rate at which it shrinks is increasing as it gets smaller and smaller and smaller. So again, because as it gets smaller and smaller, there's more energy in the system and you're trying to expel that to basically completely dissolve away the gas. So this is, if you think nature hates small bubbles, then nature really, really, really hates making new bubbles. And from the video that the moderator, Professor Nick Trefethen, mentioned his father was in and made, that I would encourage all of you to go. I put the link here. It is absolutely fascinating to watch all of the ways surface tension acts in different systems, not just in bubbles. But anyway, he made a comment that I thought really resonated with me in this talk, and it is in fact very difficult to make new bubbles. And why is that? Well, I kind of cheated in the previous video. It looks like I made new bubbles very easily. All I did was shake it. But if you notice, as this video is running, I actually didn't make any new bubbles. All I did was rearrange a currently existing bubble and mixed it into the liquid, which actually doesn't take very much energy at all. I mean, it does take some energy, but I'm not making new bubbles. I'm sort of rearranging these bubbles. And then you might say, well, actually, I make bubbles all the time. I boil my kettle, or I pour a glass of fizzy water or champagne, and bubbles seem to miraculously appear out of nowhere. So here's a glass of champagne, and you can see bubbles seemingly appearing out of nowhere. But in actuality, that's not really, again, creating new bubbles. Because if you were to zoom in, and again, in the video shows a really excellent example of this. If you were to zoom in, you'll actually notice these bubbles tend to always come from the exact same place. And that's because the glass surface isn't perfectly smooth. There are going to be cracks and crevices. And when you fill that liquid up, they don't completely wet, and you're trapping some amount of gas. And then the CO2 that's been the liquid supersaturated with CO2, that gas dissolves into this bubble, it increases its size, and then it pinches off. So you're not really, again, creating new bubbles. You're sort of rearranging the current bubbles existing in the system. So if you really wanted to create a completely new bubble, you almost need an infinite amount of energy to do it. I mean, in theory, it's roughly proportional to this. But in practice, if I wanted to nucleate a bubble out of nowhere in a pure liquid, in water, it takes almost 2,000 atmospheres of stress, of tensile stress, to then suddenly go from this top state to this bottom state. That's how difficult it is to create new bubbles. In fact, some measurements suggest that this could be even higher. It really has to do with how pure and how clean and how smooth your system is, which is very difficult to achieve. So this problem, this idea of suddenly instantaneously creating a bubble in a fluid actually was posed a very, very long time ago in, I think, the early 20th, late 19th century. And it was formulated by Besant and posed to Lord Rayleigh to find a solution to this, which basically sums up to, what would happen if you did manage to create such a bubble? What would happen to the radius of the bubble, and what happens to the pressure within the system? So he, along with some others, created this Rayleigh-Placid equation, which is the classical equation we use to look at bubble dynamics. And you can see that it's basically comprised, I wouldn't worry about all of these terms, but it's comprised of a balance of two different types of energy, loosely speaking. The energy of motion of the bubble balanced by the surface energy of the system. And you can predict, using this equation, what would happen if I did, in fact, manage to create this bubble. And what you notice is that it doesn't dissolve away like the previous equation suggested, because, again, that's a pre-existing bubble filled with gas. It's not a spontaneous formation of a cavity. What you see, it's very dynamic. It actually shrinks in size and then seems to rebound and constantly rebounds. And this actually extends for a very long time. This is in microseconds, but this continues on for seconds. And you'll notice that the pressure in the system goes from atmospheric, effectively, or vapor pressure, and then shoots up to hundreds of atmospheres pressure. In fact, there's so much inertial or motion energy in the system that if this bubble were nearby a surface, it would actually implode in on itself and hit the surface with so much force and so much energy that you can actually melt steel. And this is actually what happens in ship propellers. So as ship propellers are moving in your liquid, you're partially creating some of these bubbles. There are some bubbles already present, sort of a mixture of things. But over time, bubbles that are created that sort of implode and impact onto the surface can erode away the steel. And this is part of the motivation of why Lord Rayleigh and his colleague Plesset worked on this, solving this type of equation. So we know bubbles are very dynamic, and it turns out we have a term for this. So if we look at the formation and dynamic motion of a bubble, we call that cavitation. So in this lecture, if I use the term cavitation or I use the term bubble, I'm sort of using them interchangeably. But to be really specific about it, if we're talking about bubble motion and their formation, I am talking about cavitation. So these dynamic bubbles are cavitation, and there are many examples of cavitation, both man-made and in nature. So I'll just show some videos. All of these are taken at really high frame rates. This is one that I took in the Netherlands. It's at 10 million frames per second. This was also taken by the same group of a mantis shrimp snapping its claw, which is also, I think, around 10 million frames per second. And I think this is in tens of thousands of frames per second, but not necessarily in the millions of frames per second. So you'll notice that they all involve bubble formation. And then you can see that this behavior is extraordinarily dynamic. It's not fairly stationary. You can see how violent, especially the middle video, can be. This is what causes the pitting in some respect on the surfaces of ship propellers. And the mantis shrimp, interestingly, I mean, it's been doing it for eons, uses this as a strategy to stun its prey or break the shells of other shellfish, like clams typically, to then get at the meaty center. And so basically what I want to highlight for this is that if there are bubbles in a system, then there is useful energy to be used in that system. And if there is useful energy, then we can engineer these systems and control them for beneficial things moving for different applications. So we know cavitation can be controlled. And again, we can just take sort of hints from Rayleigh-Plesset when they worked on this equation. And we can see that there was a term at the far right that I highlighted in red that has to do with the surrounding pressure of the fluid. And we can modulate that pressure using ultrasound, which is effectively just a mechanical wave or a stress wave in a given fluid. We associate ultrasound with, of course, diagnostic imaging. If you've ever had a sonogram to see your unborn child, that's one prominent use of ultrasound. But really effectively, we're just sending out pressure waves in the system. And these bubbles present in that system, either generated or preformed, will oscillate in response to that oscillating stress. And it can do so in two ways, loosely speaking. It could do it in a non-inertial way, so mostly governed by the right hand of this equation shown above, where you can see that it kind of works out of phase with the varying pressures in the system. Or it can be governed by the left-hand side, which is more to do with the fluid inertia of the system, in which case you can get those really violent behaviors of bubble collapse or very much not dominated by the changes of pressure in the surrounding. So we know if we can control it, therefore this cavitation event is going to be useful. And what do I mean by useful? What I mean is that cavitation is able to generate a plethora of mechanical, thermal, and chemical effects, and it can actually influence everything from the micro scale all the way down to the atomic scale. So the mechanical effects you can imagine as this bubble is sort of vibrating. It perturbs the surrounding fluid, and if this persists for long enough, the fluid actually ends up convecting and creating a flow. And this microflow, also known as microstreaming, it can be on the order of hundreds of microns away from the source or even millimeters away from the source. What's also interesting is that bubbles tend to collapse in on themselves. So again, dominated by the inertia of the surrounding fluid. So that inertial cavitation or inertial event that I showed in the previous slide. As it implodes in on itself, it actually can create or generate a shock wave. And that shock wave, as it is sort of emitted spherically around that imploding event can impose its own mechanical effects and actually could, in some cases, nucleate its own cavitation events, which could implode, and you can kind of have this minor or a small chain reaction. Lastly, there are also thermal and chemical effects to consider. If we were to nucleate a bubble and it were to inertially collapse, you can see that the volumetric changes are orders of magnitude. It goes from maybe nanometers or even sub-micron in diameter, expands to hundreds of microns in diameter, so maybe 2 orders of magnitude in radius or diameter. And of course, that cubed would give you the volumetric changes. And so there's a lot of energy as it sort of implodes in on itself with those massive amounts of volumetric changes. This is a process known as, in other terms, as inertial confinement. So the vapor or gas that's in this bubble gets inertially confined in such a small volume that it tends to heat up. And the pressures are very large. I mean, we predicted with Epstein Plus that it's 400 atmospheres, but in actuality, with better models, more recent models, it can be tens of thousands of atmospheres in pressure. And the temperatures can actually reach equivalent temperatures that would occur on the surface of the sun. So these are very energetic micro or nano events. And there's so much energy that you could actually generate light. So this is a process known as sonoluminescence. Some people have called this stars in jars. If you can look it up, and you can see really interesting light shows, I guess you could put it, due to an acoustic wave. And that's due to these inertial events. But the heat in this hot gas sort of environment actually can break apart molecules homolytically, and the most prominent one, because we typically work with water, is water vapor. And so you can generate these free radicals, this hydrogen radical, which is highly reactive, and a hydroxyl radical, which is also highly reactive. And that has lots of implications of what you can do in terms of chemistry. So once again, I want to highlight that these events are so energetic because nature hates making new bubbles. And then the real question is, well, how do I create new bubbles in a system? Or what would be a better approach? So you could use ultrasound to generate 2,000 worth of atmosphere of tension and nucleate homogeneously bubbles throughout a system. That is known as inception cavitation, and people do use it for various applications. Typically, no fluid is perfectly clean. There's usually some amount of gas that's dissolved in the system. There's always some amount of debris or dirt, and those tend to act as nucleation sites, so that 2,000 atmospheres tends to be dropped to maybe hundreds of atmospheres to tens of atmospheres, depending on how dirty, so to speak, your clean water is. So in practice, most ultrasound systems can achieve that, but it is completely stochastic. It's random both spatially and temporally. You don't know when or where cavitation events occur, and they are highly energetic, so it's really hard to control. And you can imagine for some applications, in particular in health, that's not very ideal. So instead of trying to always nucleate cavitation, because it is so challenging, we tend to rely on the left-hand 2 sides of this slide here and here, using basically pre-existing or exogenous gas bubbles or gas nuclei. And so there are 2 ways we do this. Following the method of shaking a bottle, we actually basically shake a vial of liquid and create tons and tons of bubbles. Unfortunately, Epstein and Plesset, the same Plesset of Rayleigh and Plesset, showed us that these bubbles will dissolve very, very quickly, and we need them to be very small if we want to use them in various in specific applications, especially in health applications. So as I said, there are these 3 terms. Basically, these 2 terms will help tell me how fast this bubble will dissolve away. And if we could find a way to make this F term 1, and if we could find a way to make this surface tension 0, then the rate of change of the bubble will also go to 0. And the way we do this in practice is we coat the surface of bubbles with a phospholipid not too dissimilar to the lipids that form the membranes of our cells. And these do a really good job of reducing the surface tension to pretty much absolutely zero. So here's a similar video of the one that I made previously, but now it's been coated with a lipid. And what you'll see is that it resists, you can see it resisting trying to dissolve away. In fact, in order to get this system to dissolve, I actually had to degas the entire liquid in the surroundings. Otherwise, it would sit there indefinitely. And even after doing that, it actually is just like the radius time curve of that bubble. It will still persist. It's hard to see because I lost resolution, but it will persist for as long as I was willing to sit in front of the microscope to take pictures of it, which wasn't really long, actually. And what another interesting thing from an engineering point of view is these sort of lipids act as a really interesting anchor for surface chemistry. And then we could also engineer the gas composition and also play around with ways of preventing Ostwald ripening, because that actually still occurs in these systems. This type of setup on the left is used a lot in the biomedical engineering area and was the focus of my PhD at Columbia. And honestly, it won't be the center of my talk because I think it's a very well-established field. They use it in just to give you a summary of what they have used it for in biomedical engineering. These are also known as ultrasound contrast agents. So if you have a if a radiologist or sort of an ultrasound technician has difficulty imaging certain parts of your anatomy, in particular, this is used in the heart, if they can't see certain directions of flow in your heart, they will oftentimes introduce these sort of lipid-coated bubbles into your blood. And because of the bubbles' interactions with the sound wave, it acts as a contrast enhancer. In many ways, like gadolinium would for MRI or some of the other things you might consume for CT scans and PET scans, et cetera, et cetera. But then people have now used it, obviously, for drug delivery, for opening up the blood-brain barrier to enhance therapeutic diffusion into the brain, for Alzheimer's, for Parkinson's. People have used this to help facilitate drug delivery into tumors. People have now found ways to make these micro bubbles into nano-sized bubbles. They've done all sorts of interesting chemistries on the surface with prodrugs. I mean, the body of literature is absolutely massive and something I dabbled in for a good period of time, but have now transitioned to what I think is an emerging technology and which will be the focus of the talk moving forward, which is taking advantage of surface stabilized bubbles. So here you have a bubble coated by a surfactant that prevents gas diffusion. Here we actually have an inverse radius of curvature. So here, this is the radius of curvature. Here it is here in this direction, and actually helps prevent the bubble from dissolving. So this is actually more energetically favorable than dissolved gas into the system. And what we've been working on is finding clever ways of, instead of having a full surface, of creating sort of nanoparticle surfaces that might be able to trap gas. And here we can start engineering the substrate or any coatings on the surface, as well as the gas in the system. And so the work that I've been trying to pursue moving forward has largely been looking at novel approaches in nucleating cavitation using these solid substrates and engineering these solid substrates. So I'm going to focus here on just two uses of this technology. One, a medical use, and the second one being some chemistry or heterogeneous catalysis. So really quickly, I'm going to take a break here to just quickly acknowledge the team that has worked on this primarily. I have honestly done none of the, I've done very few of the experiments that we're about to see. I mostly wrote the grants that were able to fund the work. I will also quickly highlight that Summer here, who did a lot of medical work that I'm about to show, just defended her thesis last week and graduated. So she has really pioneered a lot of the work that you'll be seeing moving forward. And of course, this is not done without a copious amount of collaborators all throughout Singapore and a lot of funding sources that were in Singapore. And of course, now sort of transitioning, starting up a lab here in Oxford. So I want to look at Theranostics, which is a portmanteau of therapy and diagnostics with cavitation, with a particular interest in preventing restenosis from angioplasty, but I won't talk about that in too much detail. All you really need to know is that restenosis is not good, and it's very difficult to treat and requires long periods of time of drug delivery to treat or prevent, rather. So what do I mean by cavitation theranostics? Well, you can imagine that I have a system where or a patient that I've injected particles into, either loaded with drug or alongside a drug. It turns out that both methods would work. And I have some disease site here, and usually these disease sites are diffusion limited, as in it tends to not want drug to naturally enter into it through passive means. And so I can apply ultrasound, I can nucleate these cavitation events, which are on the order of, maybe tens to hundreds of microns in diameter. And these oscillations induce the microstreaming, which oppose shear forces, which may help permeabilize the endothelium, but also, more importantly, will help pump drug into the diseased tissue. But interestingly, if they are a solid cavitation agent, it might also pump the cavitation agents into the tissue. And once they're in the tissue, they might act as, again, further nucleation sites for either further therapy or for imaging. The way we design this is to try and trap as much gas as possible on a surface. So here we've created a polymer structure that, as you can see, has tons of pockets on the surface where gas might like to sit. It's made out of a polymer that is degradable in water, and it is it's known as polylactic glycolic acid, for those who are familiar with certain types of polymers. And this polymer is already well established in its known ability to be loaded with drugs. So we've shown that we could load it with sirolimus, which is an anti-restenotic agent, dexamethasone, which has recently, I guess, come to a lot of attention because of its potential uses in treating patients with COVID, but it's an anti-inflammatory steroid. And then we can load them with fluorescent dyes just to locate where they are. And again, we can trap gas onto these surfaces, which helps us for nucleating cavitation. And we find that we can sustain cavitation for long periods of time, which enhances our ability to either promote drug delivery or implantation of these particles into tissue, as well as enhancing the local ultrasound contrast. So what we did was we created a fake disease site by creating a transparent tissue using basically a fancy form of jelly. And we loaded this jelly with foam cell spheroids, which are linked to the basically highly inflamed tissue collection, or rather collections of cells, which better mimic, I think, what would occur in the body. It's not perfect, but it's more manageable than, I would say, and arguably in some instances more representative than using mice, because these are all human cells. And we embed this into sort of another type of tissue mimic that we can remove if needed and then analyze. And what we found was, if our particles, which I'll call SSMPs, are loaded with a fluorescent dye known as rhodamin B, which is red, or DAPI, which is blue, what we can see that if we were to only treat this with DAPI, you can see that you're reliant on the diffusion of DAPI into the foam cell. And because it's so densely packed, it tends not to reach the center, so you don't get very good treatment. With no treatment whatsoever, you see absolutely nothing. And then, of course, with ultrasound and our particles loaded with these two dyes, you can see that they've sort of proliferated throughout the entirety of the foam cell. In fact, they reside there for a fairly long amount of time. And we chose DAPI as a second dye because DAPI actually only is fluorescent when it's bound to the DNA in the nucleus, the DNA in the nucleus of the cell. And so within minutes, you can kind of see some DAPI, but it's not very prominent. And then after 48 hours, you can see that DAPI is extraordinarily prominent in the system. even though the location of the particles hasn't very much changed, suggesting that we're able to elute drug for at least 48 hours. But it can be shown that this can be done for even longer, up to 3 days, basically, with this formulation of polymer. Interestingly, if we were to load them with our dexamethasone, which is an anti-inflammatory, and these foam cells are highly inflamed, we can suppress the inflammation. And this is measured by the presence of these red droplets effectively. And so untreated, you see tons of these little red droplets, which suggests that the foam cells are inflamed and sequester or sort of secreting out cholesterol, which is what they do in this sort of hyperinflamed state. Paraphrasing a bit here, but generally that's kind of what's happening. If we provide ultrasound with our particles not loaded with the therapeutic, there's really no qualitative change. If you just have the dexamethasone, you can still see some effects. You'll notice that on the edge of the spheroid, you don't have very much red, but as you get to the core, you see lots of red. And again, that's to do with the limitations of how well the molecule can diffuse into the center of the particle, of the foam cell. And what we did notice is if you have our combined treatment, you get very little presence, at least qualitatively, of red in the system. So what about actual theranostics? So that's just the therapy side. What about the diagnostic side? So here, it's the exact same system I just described earlier. We can apply our ultrasound. We can map the presence of the particles, where we've treated. So here is a section where you have tons of spheroids located. We applied ultrasound, and what you can see is there are particles everywhere within there. Maybe some that might have migrated away because our focus is slightly larger than this region. And pre-treatment, maybe some of the foam cells might have scattered some sound, not very much. But post-treatment, you can see that suddenly it lights up, suggesting the presence of the particles. And so we can deliver both treatment and imaging and enhancement, and that's Theranostics. Right now, we're actually moving away from these foam cell systems and into the clinic, where we're working with clinicians in Singapore and working in pig models, basically, to see if we can deliver sirolimus into the arterial wall, and to, instead of treat inflammation or inflamed arteries, we're trying to basically prevent restenosis due to that inflammation. And basically, this is very preliminary data, so excuse all the very large error bars. And pigs are very expensive, and it's really hard to do a lot of them. And so basically what we've shown is our treatment method using a off-the-shelf device, catheter device, which isn't meant for drug delivery, was able to deliver our particles and sustain, and basically at a higher degree than the controls. And unfortunately, though, this is about 10 times less than what we need to get to. So we have a lot of effort now in looking at the device side. So the particles we have a good understanding of, but now we're looking at developing the device. So moving on to the second example, how can we use cavitation for catalysis, and in particular, a greener approach to heterogeneous catalysis? Well, we all are aware that the population is increasing, maybe not at such an alarming rate as in the past, but it is still increasing. And as a result, the demand for food, chemicals, and in particular energy is going to increase. Unfortunately, our current methods to produce food, which is linked to ammonia, chemicals that basically is in every aspect of our life, the plastics that we use, or the additives that we have in our food, et cetera, and even the energy that we have are all linked in some way, shape, or form to oil and gas. And this is really not going to be good if we want to create more sustainable approaches in delivering these key things for society. And so what we ideally would like is to have some renewable energy source and some biomass-derived chemical stock. So this could be sugar or celluloses, starches, et cetera, have some kind of chemical process that takes the energy from here to rearrange the molecules or atoms of this. This can be done thermally or electrolytically to either create clean fuel for energy. This could be in the form of hydrogen. So that's very prominent in sort of our modern or sort of the recent news with this green industrial revolution, where we're trying to improve the hydrogen economy of the UK. So you want to do this without reliance on methane, which is what we currently use to create hydrogen. Ideally, you could get it from water. But alternatively, there's a lot of different chemicals as well that we use in our day-to-day life, and creating the feedstock chemicals to create other types of chemicals, obtaining them from basically from nature, water and sun, wind, and I guess hydroelectric would be ideal. But the problem is these systems are both kinetically and thermodynamically limited. There's often this weird balance between kinetics and thermodynamics, which is why we are so reliant on these fossil fuels, because they're so energy dense. And that prevents a lot of sort of heterogeneous catalytic methods in obtaining these two things, fuel and chemicals. And so really, how do we overcome this? How do we supply a lot of energy to do this molecular rearranging to achieve these sorts of things? One approach that has been around for maybe 50 years is this notion of sonochemistry. And sonochemistry is interesting. You would apply ultrasound, which is basically achieved by converting electricity to mechanical stress. And the electricity can come from a renewable source. So that is inherently green. And a lot of the crystals that are used to do that transfer are very cheap and very scalable. So it's actually a really interesting low-cost solution of transferring energy around. You nucleate the bubble based on the mechanisms that we described before, and you cause this inertial collapse based on Rayleigh-Plesset. And we have this sort of adiabatic compression of a bubble, which becomes really hot and highly pressurized. Two perfect conditions for catalytic or heterogeneous catalysis. And you generate some radicals, and you'll generate some light. And what ends up happening is, if you were to like draw a box around this and look at it, is in the hot gas phase, it's so energetic that you basically break everything down. So water vapor becomes its radicals. Any volatile species that are in there might become radicals. They might interact to form different combinations. So this might get hydrogenated. This might become oxidized. Or this might become reduced. This might become oxidized. So there's all sorts of reactions going around. It's not very well controlled. And then just outside of the sort of the liquid layer, you have this hot liquid region where non-volatile compounds might get broken down because it's still hot. And it's going to be exposed to a lot of radiation from this, from the sonoluminescence. You could also have radicals that might combine to form slightly more stable hydrogen peroxide. And actually, it's been shown that you can just form hydrogen directly from the water. And then eventually in the bulk phase, where it's a lot colder and it's back at ambient pressures, you can have some of these radicals sort of diffuse out. They can react. You can create interesting types of alcohols, or you can, again, you have basically redox reactions that occur. But this is not controlled, unfortunately. It's very difficult to control. And so really what we're wondering is if there's another way. If I were to use a catalyst as the nucleation site for cavitation, then we would create this high energy environment, this sort of microreactor environment around the catalyst, thereby creating a catalytic reaction. And if I tune the materials properly, I could actually also take advantage of some of the light that's being generated and conduct photochemistry effectively. And so we're hoping we can amplify the amount of radicals generated, or even guide the chemistry that's occurring here. And so basically we've chosen a substrate similar to what the diagram I showed earlier. It's titanium dioxide, which is a known photocatalyst. And again, we have all of the necessary conditions to nucleate cavitation. And the general idea is what I showed earlier, is when we nucleate cavitation, then the light generated will basically trigger the photocatalytic aspects of titanium dioxide in combination with pyrolysis, or the hydrolytic, sonolytic degradation of the water, creates all of these interesting species that are highly reactive. And then we demonstrate how reactive it is by degrading methylene blue, which is a dye, so we can easily see its degradation. First, we check to see if these particles can cavitate, and we look at the likelihood of cavitation occurring, and we compare it to things like water or particles without gas pockets in them, and we basically see that only particles with gas pockets will cavitate. And again, it still requires quite a bit of energy. It is not, you still need energy to pull out the bubble from that pocket. And basically what we see is that we're able to rapidly degrade down the methylene blue. And in fact, we can do so by just simply ensuring that the catalytic component is in the bubble collapse. We can actually amplify the rates of degradation 3 orders of magnitude, 1,000-fold faster than all of the conventional methods. And because we're using slightly higher frequencies than other people would use, because we're already using pre-trapped bubbles in the system, we can do it at much lower power inputs or similar power inputs than other people. But again, with 3-fold or 1,000-fold increase of degradation rates. So we're wondering is, this is a very non-selective process. So can we do selective oxidation, which is more interesting if we want to create valuable chemicals? And so we basically have done the same thing, but now we've coated the particles with a catalytic site, which will help facilitate a selective oxidation process, in this case, taking benzyl alcohol and converting it to benzaldehyde. And in this system, we had to add a little bit of hydrogen peroxide in the system because we have a very closed system, and we needed additional oxygen to help facilitate this process. And so again, we have the exact same setup, same particles, and you can kind of see where these little dots are. Those are the gold palladium nanoparticles, which act as the catalytic site, partly act as the catalytic site. And what we see is that in our system, we can produce benzaldehyde 4 times faster compared to basically without ultrasound. So we just left the system to itself. And you'll notice that none of the other systems really amplify, even in the presence of ultrasound, amplify this rate. In fact, we can take the gold palladium and add it to a non-reactive surface like silicon dioxide, and it basically does absolutely nothing. So there's tons of cavitation here, but no actual reaction. So there is this engineering of the surface is quite an important process to this. And our work compared to other people's work, unfortunately, well, this is a more challenging reaction. So we weren't able to get that 1,000-fold improvement. But what we were able to achieve is orders of magnitude less energy necessary to do the reaction. And that all has a lot to do with just preceding the gas into the system. But we're able to achieve comparable rates compared to other types of systems. I would ignore these. These seem like these do really, really well, but they use what is known as a homogeneous catalyst, which has amazing benefits, but are huge costs in separating and recovering your product. So in conclusion, basically, we're engineering these bubble traps so we can overcome nature's hatred for creating new bubbles. And then we're taking nature's hatred for bubbles and using them as energetic sites to do all sorts of interesting things, such as drug delivery, diagnostic imaging, or even selective chemistry. So with that, I'd be happy to answer any questions that people might have, and looking how we take sort of inspiration from champagne, using it to create particles, and then ultimately to do something with that. So yeah, thanks a lot for sticking with me for this journey.